Okay, so we were talking about the treatment options of using the cells and materials from the immune cells, and this is what we call as cancer immunotherapy. Immune system or our immune cells can be harvested in different ways, can be utilized in different ways. One of the ways that we can harvest and create antibodies. Antibodies are specialized molecules which are designed to capture a particular antigen. They not only capture the antigen, but they can also have the potential to destroy the antigen as well with the process called complementation, antigen antibody reaction. This is one thing. This is one way we can use our immune system. The other way is that, which is quite futuristic, to creating cancer vaccines. You will see the complexities and the troubles which are associated with cancer vaccination. And the last thing is that we can activate our immune system to boost our immune system so that they're able to detect cancer more easily. It's like non-specific immunotherapy, helping our immune system to stay on alert, to stay activated, so that they're able to fix and find the bad things. Okay, there are different, the next slide completes everything, but before I go to the next slide, I want a space to write in there. This slide will explain all the possible ways we can use our immune system, all the possible ways we can use our immune cells to kill the cancer, all potential means. One slide explaining everything. अहमद नईम ने मुझे बताया कि 3 मिनट रह गए हैं. अहमद, अगर 3 मिनट रह गए हैं मेरी जान, तो आप हमें, of course the meeting will be over, okay, and you will use the same link to reconnect with me once again. Okay, let's start with the possible options. The first is that how we can use our immune system. The first thing I told you is that we can use antibodies. There is a term called monoclonal antibodies. Monoclonal antibodies means the antibodies which are specifically created against one particular antigen. And all the antibody molecules are same. And all the antibody molecules are directing or targeting one particular antigen. इसको मैंने डिटेल से molecular immunology के lectures में explain किया हुआ है. You don't need to understand that thing at this point. But if you want to know what is the difference between monoclonal and polyclonal antibodies, you can try to find the answers from there. But they are antibodies which are designed against specific antigens. If you able to create an antibody specifically against a tumor cell, against a cancer cell, look at the color of this antibody. Just note down the color and look at the color of this tumor cell. So that means these antibodies can distinguish specifically the tumor cells. Don't forget, there exist normal cells out there. There exist normal cells out there. So these antibodies will not going to bind themselves with the normal cells. If somehow you're able to generate such antibodies which can recognize the tumor cells, these antibodies will going to bind on the surface of the tumor cells, and they can create the immune reaction, and they can kill cancer cells specifically. Now the big question is that how we can create an antibody which can specifically detect the cancer cells. So we should have something which is specifically produced within the cancer cell. If there is a protein structure which is specifically created in the cancer cells, and that structure is not there in the normal cells, this unique thing can be harvested, can be utilized to create those antibodies. If you don't have any specific difference between the normal and the cancer cells, you will never be able to create such antibodies. It's very rare. to create an antibody, because it is very rare to find a specific difference between the normal and the cancer cells. I'll tell you why, and that explains a lot of other questions as well. Okay guys, so here we go. If in case we able to detect one specific thing which is present in the tumor cells and not present in the normal cell, we might be able to generate such type of monoclonal antibodies. तो एक खांसी का सिरप ले ले,
सर तबीयत बहुत खराब हो गई है।
अल्लाह ताला तेरी मदद करे, तेरा इलाज करते हैं, ठहर जा जरा, सारे मिलके करते हैं। पहले मैं इसको फिक्स कर दूं, ये बहुत जरूरी बातें हैं, खतरनाक बातें हैं। Okay guys, so having specific antibodies which can successfully detect tumor cells as compared to the normal cell is very rare, because you need something which is uniquely present only in the tumor cell. And that is very rare. Until unless the cancer cell become very different, until unless the cancer cell become very weird, until unless the cancer cell is started producing very strange type of proteins, you will never be able to get this type of antibodies. But if somebody is lucky enough, and they find अभी लोग आ रहे हैं, सॉरी अभी और लोग आ रहे हैं। But if someone is lucky enough,
if the cancer is slightly different, wouldn't the body already detect it?
Yeah, it's very rare that the immune system is able to detect the cancer. The immune system is able to detect the cancer when the cancer started producing a lot of weird things, not before that. Before that, it is almost like similar to the normal cells. And if we have something which is only present in the tumor cell and not present in the normal cell, only under these conditions, a monoclonal antibodies against the tumor cell can be raised. But maybe someone is lucky enough. Otherwise, you should remember that cancer cell arises from the normal cell. The genetic code, the DNA is same. Slight mutations are out there. The protein structures present in the cell membrane, they are same. So how the T cell or the immune cells will be able to figure out the difference between these two? Until unless this thing is very, very different. Until unless this structure is very, very different. Until unless this thing is started producing a lot of weird things, the immune cells will never be able to detect. Immune cells will only be able to detect cancer with two things. Number one, the immune cells should be high in numbers. For example, if the probability of detecting the cancer is 0.1%, how about sending more immune cells to the site? In that case, the probability of detection will be increased. That is like increasing the number of immune cells. So this is one thing. The second thing is that how about increasing the number of those immune cells which are able to detect the cancer cells? So immune cells, this one is not able to detect. Think about the immune cells. There's another immune cell that is very smart. That immune cell is very smart. That immune cell is able to detect this is the bad thing. Now, do we need these type of immune cells which are not able to distinguish, or do we need this type of immune cell? So this type of immune cell is important. So people can use this type of immune cells to kill the cancer cells. I will tell this thing a little bit later, but let me share this thing first. So monoclonal antibodies. The other use of monoclonal antibodies is to stop the immune cells checkpoints. If you can remember, I told you that there are certain things present on the immune cells. The T cells are carrying CTLA4. This is one of the receptor. It carries PD1. This is another side of receptors. These things help the T cells to sense that the other member is a normal cell. The other member belongs to us. So these antibodies, for example, the green one will go and bind with the CTLA4. We can raise antibodies against the CTLA4 receptor. We can create and raise the antibodies against the PD1 domain. This is another receptor. This one can go and bind with this one. We can create another antibody, still another antibody, which against the PD1, PDL1 domain, but this is present on the normal cells. We can block this one as well. These receptors are the immune system checkpoints. These receptors are responsible to stop the immune cells not to fight against the other member. If you block these receptors, that means T cells will going to be an indiscriminate killer. The T cell will going to kill everything, whatever comes in front of it. Whether it is a normal cell, it's a cancer. That was the question exactly asked by one of the students. किसने पूछा था ये सवाल? शाबाश इसको मैंने. हाँ? वो जाग जाओ. क्या नाम है आपका?
Sir, Zunaira.
हाँ, जुनैरा ने सवाल पूछा था. So the T cells will become an indiscriminate killer. ये एक cost है जो हमें देनी पड़ेगी, because you are blocking the checkpoints which blinded the T cells, and it will not be able to distinguish between the normal cell. It will going to kill this one. The answer is yes. It will going to kill this one. Now the T cells become blinded. It's a cost. You can take this risk. हाँ, कैसे कम कर सकते हैं? You can block, you can send the drug, for example, you can send those antibodies right in the center of the tumor. For example, there is a tumor in the body, there is a tumor in the tumor cells in the body here. So this is the tumor site. All the other sites, normal. This is the tumor site. You drop those antibodies right in the center of the tumor. You drop those antibodies right in the center of the tumor. Indiscriminate, and it will going to create all the immune cells, whatever the immune cells are present there. These antibodies will convert them into indiscriminate killer. So if you drop those monoclonal antibodies right in the heart of the tumor, all the immune cells out there will become indiscriminate killer, and they will start destroying whatever things present outside. And you can ask me, sir, वो तो tumor cells के अलावा normal cells को भी मार देंगे. The answer is yes, मार देंगे, of course. But since you are putting it right in the tumor, so I'm assuming it will kill more normal cells. It will kill more tumor cells than the normal cells. That's the one. Okay. The other thing is that you can teach. Whenever something bad happens to our body, whenever a pathogen enters into our body, our immune cells capture that pathogen. Our immune cells capture that pathogens. They try to read, they try to think, okay, what is that stuff? And then they communicate that information. They communicate that information about the bad thing to the next fighters, to the next cells, the real cells, and they are called as the T cells. How about you can help the immune cells not to worry. You can educate the immune cells about the cancer. You can generate some tumor peptides. These tumor peptides, these proteins, these receptors, they are specifically present within the tumor. You can get some peptide from the tumor cells, and you can help and educate the immune cells. Remember, these peptides are supposed to be captured by the immune cells. This is one of the member of the immune cell. It's called dendritic cell. It will capture the antigen, and from where these antigen come? These antigen came from the tumor cells. You have created, you have sent it. The dendritic cell will going to capture this, and they will going to communicate that information to the T cells, and that you can see here. and that will activate the T cells, which can kill the tumor cells specifically. It's very interesting. It's doable. Efficiency is less, but that's wonderful. The other thing is that how about ये treatment हुआ है एक patient. How about extracting the T cells and the dendritic cells? How about extracting the immune cells out of the body of the patient and out of the tumor of the patient? How about extracting this thing? This is the one who knows. This is the one who knows. How about extracting the immune cells from the tumor, multiply them, increase their number, or engineer them? ये वाला word मैंने भी अभी कहीं पढ़ाया नहीं आपको. Engineer them. The first thing, multiply them, increase their number, and then put back into the person's body so that it can specifically to increase its number so that it can effectively kill the cancer cells. And there are other things as well, which is called as biological response modifier. So there are a lot of things we can try in order to kill the cancer cells using the resources present in our immune system. This is a slide which you might be able to understand now more easily and quickly, that how the peptide from the tumor can help in the generation of the immune cells to kill the cancer. This is like using our immune cells, and in an easier word, it's an immunotherapy. So extracting the immature dendritic cells from the patient's body, from the patient's blood, get the tumor from the patient's blood as well. The person is not able to kill the cancer. The person's immune system is not able to recognize the cancer. That's the problem. So you get the tumor cells. You break down the tumor cells into different small peptides. You mix them with the dendritic cells. You help the dendritic cells to know about the tumor cells. You can engineer it as well. I can tell you. I mean, you can send using the vectors. You can send the peptides. You can do multiple things. All you need to do is to familiarize the dendritic cells about the tumor. The most easiest way, create a cell lysate, mix them with the dendritic cells so they're able to figure it out. But that the other thing is that send the vectors, the other thing, send the tumor peptides. So use all these ways to educate the dendritic cells. And now the dendritic cells are educated, they know about the tumor. Using those dendritic cells, so they can communicate that information about the bad thing with the T cells. You can drop these things right nearby the tumor. For example, in case of brain tumor, if the CTLs are there, if the T cells are there, the dendritic cells will going to activate the, remember, the dendritic cells, the DC cells will going to talk with the T cells. Will going to activate the T cells. The T cells got activated. Activated against the information you have installed. That T cells will become CTL. CTL is called as cytotoxic T cells. These cells are able to kill the other cells. Now these CTLs will be able to kill all the cells which are carrying these antigens. And these antigens came from the cancer cells. So the CTLs will selectively start killing the cancer cells. That's one interesting way how to kill things. Yes, okay. मैं 2-3 मिनट में आपको explain करता हूँ कि कैसे, how it works. Okay, how it works. Let's start. So if something is entering into our body and that something is not belong to us, that may be a cancer cell, that may be a bacteria, pathogen. Let's suppose it's a cancer cell. We are studying cancer immunotherapy. It's a cancer cell. The cancer cell is like a 99% human cell. There is a 1% difference in its structure. There's only a 1% difference in the structure. 1% of proteins and DNA and other molecules might be different. If you understand this 1%, that's a wonderful information. You can break down this cancer cell and then you can find out which of those 1% are different. You can extract those 1% out of the cancer cells and you use that 1% different thing present in the cancer cell to educate the dendritic cell, because the job of the dendritic cell is to capture the antigens. And what are the antigens? This is an antigen. What is the special thing about this antigen? Because this antigen belongs to cancer. It does not belong to the normal cell. The dendritic cells will be able to capture this thing. They will be able to eat this thing up. And after that, they will going to communicate this thing. In the next phase, the dendritic cell will going to communicate this information with the next member of the immune system, which is called as the T cell. Okay, the dendritic cell is carrying this thing. And you know what is that thing? That thing is the antigen coming from the cancer. Here, this is the place where the communication occurred between these two things. This is the place where the communication occurred between these two. The T cells got activated, and after the activation, they produce a lot of things, and one of the cell is called as cytotoxic T cells, or called as CTL. Now this CTL has the potential. This CTL has the potential to kill another cell. But what type of another cell? This has the potential to kill another cell, but another cell which is carrying the antigens. Now this is the cancer cell, and most of the cancer cell is made up of proteins which are somehow similar to normal cells, because it is produced from the normal cells. Most of the material, so like normal cell material is like 99%, cancer cell material is about 1%, it's like that. But this CTL is designed to kill anything which is carrying a cell which is carrying the antigen, this antigen, because it is raised against this antigen. Education starts from here, raised against this antigen. Now this CTL is specific killer. It's a specific killer. It will kill all the cells, and you know who are those cells? They are the cancer cells. They will going to kill the cancer cells which are carrying this antigen. Once they found it, बताइए क्या करेंगे? They will start releasing the chemicals, and those chemicals will destroy and make it a blob of this thing, and it will destroy the cells. Now imagine if naturally the dendritic cell is able to detect the cancer cell naturally. What will happen? The dendritic cell will going to communicate that information naturally with the T cells. T cells will going to produce the CTLs, and CTLs will going to kill the cancer cells. It happens most of the time. But what if they fail to detect it? In that case, the tumor cells will grow, and it will end up in cancer. What you can do, you can help the dendritic cells. How? You can give the dendritic cells the specific protein, so it get the right education and activate and create the CTLs. So one way. You can increase the number of the tumor cells as well. You can increase the number of, you can increase the number of T cells as well. So all these things you can do. You got it?
सर आपने जो बताया कि मोस्ट ऑफ द टाइम डिटेक्ट हो जाते हैं ये जो कैंसर सेल्स हैं,
हो जाते हैं, तभी तो हमें कैंसर नहीं होता.
अच्छा तब नहीं होता, ठीक है.